Yeah.
Yeah, I can tell you. Oh, it was fine.
We're doing some workshops.
Yes.
All right, morning, everyone. Monday morning, so wake up. So it's my pleasure to introduce Dr. Adam Wende to the Institute. Adam is Associate Professor in the Division of Molecular and Cellular Pathology and Department of Pathology at the Heersink School of Medicine at the University of Alabama in Birmingham, UAB. So he's an international leader in cardiac metabolism. He trained with Dan Kelly to do his Ph.D. and did postdoc with Dale Abel at University of Utah, where he got to know Junko. They were colleagues, compatriots, friends together there, and did many things like attend meetings and work together. So it's great to have that personal connection. Also, when Dr. Wende is here, he's going to be doing some workshops on animal models, mouse models of human disease. And so we're really grateful for that help, Adam. It's great. So without further ado, he's going to talk to us today about O-GlcNAc modifications in cardiac hypertrophy and heart failure, particularly as it relates to diabetes. And so at this point, I'll turn it over to you, Adam.
Okay. Everybody hear me? Perfect. Thank you for that kind introduction, and thank you for the invite. This is my first time to Roanoke, and I've enjoyed it so far. Very beautiful scenery, and I've had already several great conversations to start out the day, so I'm looking forward to the rest of it. And as far as the workshop goes, I'll describe that in a little more detail as we start going through the data. So I've broken this talk down into three main parts, and as far as my presentation style, I definitely prefer to be interrupted. So if you do have any questions throughout, please just ask them as we go, and don't save them all for the end. So the first part's just going to be my take on cardiac metabolism, why I've spent the last 23 years studying it, and why I think it's still important, because there have been people studying it a lot longer than that. But then get into some of the mechanisms of how we think alterations in cardiac metabolism impact disease progression, particularly in the context of diabetes, and particularly in the context of glucose utilization pathways, not only for glucose as a substrate for metabolism, but also for glucose as a substrate for signaling pathways. And then I'll finish off, if time allows, to get into some of these alternative mechanisms that we think glucose signaling is involved in. Now, I don't think this audience needs to all the introduction that I have, so I'll try and go through this pretty quickly, but we all know that diabetes has been a pandemic long before COVID, and that it's been increasing at alarming rates with projected triple-digit percentile increases in some regions of the world in the next 25 years or so. But what we also know through a lot of epidemiological studies is what some of these risk factors mean to cardiovascular health. And one of the reasons why I ended up in Alabama, and pardon my voice, I was on a field trip with my daughter and maybe did a little bit too much screaming on the roller coasters. Is that Alabama, unfortunately, is leader in the country for several of these risk factors. We're routinely in the top 3 for diabetes, obesity, inactivity, and then the associated hypertension and heart disease. And for comparison, I did look up your guys' numbers, and you're doing much better than we are. And I think the worst is diabetes at 21% or 21st percentile. And I guess hypertension kind of tracks with there. But what we also know from all this is that diabetes does disproportionately increase the risk from cardiovascular diseases. And a lot of people have been looking into this and what are the mechanisms, and we are one of those groups. Now, there's a lot of other mechanisms that go on in parallel with the GlcNAc theme that I'm going to tell you about today. I don't think that metabolism is the only mechanism, but it is what we've decided to focus on. So that's what I'm going to say. most of the focus is on, but I do think that this works in concert with many of the other pathways that several of the labs in this room are working on, as well as those across the world. But what got me really interested in the idea of cardiac energy metabolism really impacting the disease state is the heart's unique ability to utilize so many different fuels to meet its constant contractile needs, of the idea that the heart needs to beat to get the nutrients to flow to other organs. And because of this, I think evolutionarily, the heart's adapted the unique ability to be what's often referred to as a metabolic omnivore, using whatever substrate it is provided in a concentration dependent manner, with fatty acids, particularly in mammals, being one of the most fuel efficient ways. And oftentimes glucose being a secondary metabolism. And as we've seen over the last 5 to 10 years, other metabolites have definitely taken center stage in how their regulation may be impacted in disease state. And I'll get to some of that in a moment. But this is not a new concept. So going back to the early to mid 50s, people were looking in both canines and humans in the diabetic state to see how the heart could use these two primary fuels. And what was found was that there was a paradox of that although there was higher circulating glucose around, those hearts would preferentially increase fatty acid utilization, but in an uncoupled manner that often led to toxic lipid intermediates or inefficient ATP production. What I didn't know, though, until starting my faculty position, was that all this early work was actually done in Birmingham before UAB was actually even founded. So kind of coming full circle for my career. So this has led us and many others to look at what happens to cardiac metabolism in different disease states, highlighted here in the context of the diabetic heart, where despite this higher circulating glucose, there's changes in insulin signaling, changes in glucose signaling, changes in lipid signaling that all lead to decreased metabolic efficiency. And one of the big keys in there is what's going on with this decrease in glucose utilization. Is this a maladaptive response to hyperglycemia in the diabetic states? And that is exactly the first question we set out to ask. So is this change in cardiac metabolic substrate flexibility, or the ability of the heart to use this excess glucose loss, is that maladaptive? So I try to go quickly through the background since this is a cardiovascular audience. So hopefully none of that was new. And if it was, I'm sorry if I went too quickly. But what led us to this idea or question of if the loss of glucose utilization was adaptive or maladaptive came from studies a little over 20 years ago now by Daryl Belke and colleagues. And what this group did was in a DB-DB model of diabetes, looked at cardiac substrate metabolism. These are results from isolated working heart preps where they perfused the heart with radio-labeled glucose and measured the glycolysis rates and glucose oxidation rates in either control non-diabetic mice or diabetic mice. And what they saw, consistent with some of that work in the 50s, is that glucose utilization went down in both cases. And they also had this similar hypothesis that maybe enhancing glucose uptake or restoring the glucose utilization could protect this. So what they did was they overexpressed the glucose transporter GLUT4 systemically in those animals and showed that they could rescue the glycolytic and the glucose oxidation of those hearts, kind of setting the premise of the question that we sought out to answer. However, there were a couple caveats with this model, namely one being that the GLUT4 was overexpressed from birth and it was overexpressed systemically. So the initial hypothesis that kind of started my career going in was, could restoring myocardial specific glucose delivery attenuate or protect the heart from the development of diabetic cardiac dysfunction, and not only the form of metabolic derangement, but also some of the contractile dysfunction that accompanies it. And to accomplish this, we use the DOX-ON system or TET-ON system, which I think is pretty commonly used now. But briefly, this is a double transgenic model where we confer cardiomyocyte-specific expression using the alpha-myosin heavy chain of the transactivator protein, which in the absence of its ligand floats around in the cardiomyocytes. but in the presence of DOX can bind to a TET response element and induce whatever transgene is behind that promoter. And in our case, it's a MIC-tagged GLUT4. So this model worked. This is just a Western blot showing that in control single transgenic mice, either in the absence or presence of doxycycline, there's none of the MIC-tag. And even with both transgenes in the absence of ligand, there's no MIC-tag. But once we have both transgenes and doxycycline, it's induced. Most people know GLUT4 for its insulin responsiveness. In the heart, that is part of its mechanism, but GLUT4 can also translocate to the cell surface in response to calcium and contraction. So in the heart, it's a little bit different than skeletal muscle, but we can still visualize that and show that in isolated cardiomyocytes that in response to insulin, the exogenous MIC-tag GLUT4 goes to the surface. And this corresponds to an enhancement of glucose uptake. So the model works. But could we use it to rescue diabetic cardiomyopathy? So to test this, what we did was we took a cohort of animals, either single transgenic or double transgenic, and treated them with either vehicle or streptozotocin, which is a beta cell toxin, to initiate an insulin deficient type 1-like diabetic state. But we use it primarily just to make the mice hyperglycemic. So to get to this question of if the heart's exposed to excess glucose, what would happen? And then once the mice were already diabetic, that's when we then induced the transgene and then followed the mice out for a couple more weeks. And all the data I'm going to show you for the rest of this model was done at this 4-week time point where the mice were, yes.
Sorry, I've been running across this in my own research as well, but it's the use of these models when we call them diabetic.
Yeah.
But we get into the 500 to 600 range of these measurements. What are your thoughts on that? Because I had a med student come to my lab as we were doing these experiments, calling him diabetic. She was like, "That's not diabetes, that's glycemic shock."
Well,
she's like, "This is like trauma."
Yes.
To these animals, she's like shocked that they were even like walking around the cage, like seemingly fine. She's like, "These animals should go to the ER." I was like, "Well."
Yeah, so the streptozotocin model is not perfect. We've actually moved away from it after this project. I think this will probably be my last project using it. And my response to that is it would be diabetes 110 years ago before the discovery of insulin, right? So this is untreated diabetics, which would go into glycemic shock and die. But now we have insulin treatment. So some people will use STZ and then treat with insulin to keep it in a more manageable rate range. But that's also part of the reason why this is only a 4-week study instead of a longer study. What we've moved to, and I'll show, I think, one data point from, is this new combined high-fat diet, very low-dose STZ, where they still secrete some insulin, but they have more of a diabetic-like phenotype. Because the other thing, too, is often in the literature, STZ is called type 1 diabetes, but type 1 diabetes is an autoimmune disease that this has no component of. Yeah.
I'm just curious about that. I for a short while was submitting grants on this and kept getting dinged on certain things, and not that necessarily within my med student question. So I'm just curious about your thoughts.
So we did do a review. John Usher is the main person who led it in AJP, I think 2 years ago, going in the different diabetic models. So you can, I would refer you to that. For no model is perfect, especially.
Well, it's just one of those things about semantics. We call it certain models diabetic. What is it?
Exactly. So, and that's why STZ induced hyperglycemia.
There you go.
So, and that's exactly what we're looking for. So could we get these mice hyperglycemic, and then what would happen? So shown here is that same isolated working heart prep. And first off, just looking at the vehicle treated mice, where when you enhance GLUT4, you can see enhanced glucose uptake and glycolysis and enhanced glucose oxidation. Similar to what was shown in the Belke study, we could rescue the defect in glycolysis. However, unlike the Belke study, we actually exacerbated the glucose oxidation defect. So this led me to revisit my original hypothesis. And not only did it appear that enhancing or restoring glucose delivery to the diabetic heart does not attenuate, it might actually enhance development of diabetic cardiac dysfunction. And all that was to set the stage for why O-GlcNAcylation. So this led to a big theme shift in my mind and in the project in general, of if it's not nutrient delivery for metabolism, what else was the glucose doing? And that led me to this pathway called the hexosamine biosynthesis pathway that leads to the generation of UDP-GlcNAc, which is used for protein glycanylation. And I assume since Junko's on GlcNAc, you now are familiar with it to some extent. Basically, the way I describe it to people is everything you know about phosphorylation is also true for GlcNAcylation. Except for the fact that when you think about phosphorylation, you're dealing with hundreds of kinases and phosphatases to regulate its various processes, where with GlcNAcylation, there's only one gene, O-GlcNAc transferase, to add GlcNAc, and one gene, O-GlcNAcase, to remove GlcNAc. So this has led several people, including myself, to postulate that it's primarily driven by nutrient signaling then and stress responses. And this has been shown to be regulated in several physiologic conditions as well as pathologic conditions. So with this as the context of if glucose wasn't simply going to glucose oxidation when it was being enhanced delivery in this diabetic state, where might it be going? And if it was going to GlcNAc, could we find that? So we did a Western blot for just pan-GlcNAcylation. Also, unlike phosphorylation, there's only a handful of GlcNAc site-specific antibodies. So the general tools for looking at it in the field are to use just a pan-GlcNAc antibody. There are two primarily used, and just look at all the sites of modification. And what I hope you can see here is that enhancing glucose delivery in the MICGLU4 mouse could enhance protein glycanylation, and diabetes further exacerbated this. So because we don't have site-specific antibodies, the way we get to protein identification in this case is we did a 2D page and then did a staining for glycosylation and picked the sites that were the spots that were most differentially glycosylated, and performed mass spec, and found that of those that had the most enhanced glycanylation, almost all of them were involved in oxidative phosphorylation, kind of consistent with this idea of the decreased glucose oxidation despite the enhanced glycolysis. So there appears to be a protection of the heart in decreasing glucose uptake in the context of hyperglycemia, because a lot of these sites by other groups have also been shown to diminish activity, particularly in the OXPHOS complexes. But I'm actually a transcriptional biologist by training, so all this protein work is interesting to me, but a little bit outside my wheelhouse. So we also did transcriptomics in these animals with the intent of defining, is glucose only signaling to post-translational modifications of these enzymes, or is it also signaling to other cellular pathways? Because when GlcNAc was initially discovered as a signaling molecule, it was discovered on transcription factors to regulate gene expression. So we performed transcriptomics in these different cohorts to try and determine what of the changes in diabetes were specifically being driven by enhanced glucose. So shown here is a just Venn diagram of diabetic mice, shown here in the bottom right, GLU4 overexpressing mice, and the combination of the two, with a focus on what genes were being upregulated in all three conditions or downregulated in all three conditions. And what we found, again, consistent with the post-translational modification of the proteins, the gene expression was also consistent with metabolic regulators such as pyruvate dehydrogenase kinase and beta hydroxy dehydrogenase, suggesting that glucose alone is sufficient to drive a lot of the changes in metabolism. But this still was only guilt by association. We wanted to try and determine if there was any direct regulation. So what we did was cloned out one of the subunits of complex 1 that was downregulated in our models into a firefly luciferase vector and transfected it into cardiomyocytes, H9C2s, and then treated those cells with either low glucose or high glucose to simulate our two conditions. And what's shown here is a schematic of the promoter region that's conserved between mouse and human at a few specific regions. And the conserved regions had response elements predicted to bind the transcription factor SP1. So we did a deletion series showing that high glucose could suppress promoter activity in 2 kb upstream of the transcription start site, 500 base pairs, but that was lost 300 base pairs. And within that 500 to 300 base pair was one of these response elements that when we mutated it, completely lost glucose sensitivity. And then to show that this was specifically through the transcription factor SP1, we overexpressed SP1 exogenously and showed that this further enhanced the suppression by glucose. Now, how does this relate to O-GlcNAcylation? So as I alluded to a moment ago, one of the first targets of O-GlcNAcylation were transcription factors, and one of those were SP1. That was part of the reason why we used that as a candidate approach in the promoter studies. So what we wanted to test was GlcNAc driving this change? So using those same luciferase reporter assays, again, showing high glucose could suppress promoter activity, overexpression of green fluorescent protein had no effect, but overexpression of O-GlcNAcase, which would remove GlcNAc, could attenuate or prevent that glucose-mediated suppression. And through immunoprecipitation, we confirmed prior data showing that SP1 in these various models was indeed GlcNAcylated. So since this is already published a few years ago, I'm only choosing a few snippets of the data. But what this led us to conclude and was summarized nicely by this accompanying editorial was that despite our initial hypothesis, the healthy cardiomyocyte does preferentially use fatty acids, but can switch to glucose readily. In the context of hyperglycemia associated with diabetes, the heart actually appears to be reducing glucose uptake and utilization to protect itself from these other auxiliary pathways, because when we circumvented that transgenically, we actually exacerbated the diabetic-like phenotype and led to what we think is a glucose toxicity, strongly suggesting that targeting this pathway, at least to cardiomyocytes, might not be the best way to go. Now, in context, going back to the 2000 Belke paper, one of the reasons why we think that did work is A, it was being overexpressed before diabetes, and B, since it was being overexpressed systemically, the skeletal muscle was able to be a glucose sink, so that the circulating level of blood glucose in those mice actually went down, whereas in our model, since it was cardiac specific, it did not go down. And also getting to the question that was raised, it could just be that because this was a super hyperglycemic, that this might have not worked, and doing it in a type 2 diabetes model might show more promise.
And can I ask,
Definitely.
where are the enzymes localized? So you have this, whatever O-GlcNAcylation is occurring in the mitochondria and in the nucleus.
So great question. So if you go back to the early GlcNAc literature, it was thought to only be nuclear cytosolic. And then in about 2009, a series of papers came out showing that it is also directly in mitochondrial. So I introduced the pathway pretty simplistically, that there's one enzyme to put it on, one enzyme to take it off. There are splice variants of each of those that do have subcellular distribution. And John Chatham, who's a collaborator on a lot of this work, and I have an R21 where we're going to try and do some confocal microscopy to tease that out with some tagged versions of it. But that is a very good point of where it's occurring, because one of the big questions early on, particularly for the mitochondrial O-GlcNAcylation, was how is the UDP GlcNAc getting in? And that was also discovered, I think, about 2014. It was a diabetes paper where there is a translocator to get the UDP GlcNAc in there for localized O-GlcNAcylation as well.
And then my second question is, a lot of the targets that you're seeing, whether it be transcriptional or just the O-GlcNAc levels, are like TCA cycloenzymes, electron transport chain, and tying back to what you saw with glucose oxidation, do you find that it's the entry point, or do you think it's like controlling glucose oxidation and then it's kind of feeding back and preventing glucose oxidation from occurring?
That's a really good question. I think based on some of the oxphos complex activity, I think a lot of it is occurring right at that level. So Wolfgang Dillmann did a series of studies focused on O-GlcNAcylation of the individual complexes, where he showed a decrease in activity in a diabetes model, and using the same OGA overexpression could restore oxphos activity. But I didn't look in that context because we're still seeing enhanced glucose uptake in this model, and we're still seeing enhanced glycolysis. So in the cytosol, everything still seems to be working normally, but good questions. So that's it for that story. Yes.
I have a couple of questions.
Of course.
So the overexpression of GLUT4 increases delivery uptake of glucose into the cell, and yet it was not used in glucose oxidation. What is the bottleneck? Is it the hexakinase or else?
It's a good question. So we did look at glycogen storage. So some of it's going to glycogen storage. And then we think some of it's being shunted into the hexosamine biosynthesis pathway, but we haven't done the tracer studies to prove that. But what we think is that, at least what I would postulate from some of the data I'm not showing, is that the glucose is still getting through glycolysis, maybe even getting into the mitochondria to get to Jess's point, but then building up there and leading to all this accumulation of the modifications of the proteins themselves.
And a related question. So you of course mentioned one pathway, but there is also polyol pathway,
Yes.
and pentose phosphate shunt. How they what is their relative role?
Yeah, I did look at those. I don't think there were changes, or otherwise I would remember, because I have been asked that question before. I should include it, but I do not think there were robust changes in those pathways. We would predict there would be, especially in the PPP, but I don't remember there being anything that stood out. But good questions, thank you. So one of the other things I wanted to highlight, especially for the trainees, is letting the data lead the way. So I often tell the people in my lab that most of my grants and publications have actually come from when I was wrong with my hypothesis, because when I was right, anybody else could have thought of it as well. So similar to this one with the focus of thinking GLUT4 overexpression would protect the hearts, that not, is also the idea of, well, what were some of these other metabolites? So my first postdoc, Manoja Brahma, was looking at the data and noticed that one of the key enzymes that was downregulated was BDH1, which is this beta hydroxybutyrate dehydrogenase, which is one of the rate limiting steps in ketone body metabolisms. And at about the same time of realizing this came a study looking at some of the SGLT2 inhibitors and how they might be working, and it could be working through ketone body metabolism, which was the opposite of what was happening in ketone body metabolism in non-diabetic hearts. So there seemed to be this metabolic switch, which I didn't get into in this introduction, of the difference between non-diabetic heart failure, which often appears to be more of a reversion to the fetal glycolytic phenotype, whereas in the diabetic heart failure seems to be more this disruption of lipotoxicity. So Brahma was curious about this and curious to see, was GlcNAc driving this ketone body metabolism, and might this be part of the driving difference between a diabetic cardiomyopathy and a non-diabetic cardiomyopathy? So that's repeated here. So he went in to test this, saying within diabetes and this hyperglycemia, driving a change in O-GlcNAcylation, would it also be regulating ketone oxidation? And could this also be impacting the overall energy status of the heart? So to first test this, he took advantage of some of our data sets. Two I've already introduced to you, the GLUT4 overexpressing heart and just the STZ diabetic heart. And then we also had the advantage of having a small subset of human cardiac biopsies collected during left ventricular assist device implantation, where we performed transcriptomics on, specifically looking at those that were diabetic and non-diabetic, and then said within these three models, what were the common gene expression changes? And there weren't many in this initial study. I think this was actually using our original array data. We subsequently expanded it to some RNA-seq. But fortunately for Brahma, the top pathway that came out was ketone body metabolism, and then going specifically focused at the human diabetic samples, BDH1 was also suppressed in the diabetic cardiomyopathy. So now we wanted to test, was this directly impacting ketone metabolism in these hearts, specifically glucose-driven? So this time we teamed up with Martin Yun, who's an investigator at UAB with me in the cardiology department, and performed those same tracer studies and found that indeed enhancing glucose delivery, again, enhanced glycolysis. This is a confirmation of the earlier studies, But what we added in this study was to also use radio-labeled ketone bodies and show that in the context of this enhanced glucose utilization, there was a suppression of ketone body utilization. But was this being driven by GlcNAc, and how could we prove this? So what Brahma did was looked across several of our models, the STZ model, that high-fat diet, very low-dose STZ, and the GLUT4 overexpressor, and showed that in each of these there was modest to significant enhancement of O-GlcNAcylation. But this was still all correlation. This was no direct regulation of GlcNAc2 ketone body metabolism. So to specifically ask that question, we went to the old literature, and there was a rat that was identified back in the early to mid 70s in Japan, the Goto Kakizaki rat, that had spontaneous diabetes. And subsequent studies found that part of this spontaneous diabetes may be because of a splice variant in O-GlcNAcase. And that splice variant is shown here in the schematic, where exon 8 is missing. And in these two diabetic prone models, that's the exon that contains the GlcNAcase activity, suggesting that this could lead to enhancement of GlcNAcylation. So Andy Peterson in the Lake Kudlow's laboratory took that cDNA and generated a mouse where they overexpressed this, what was referred to as a dominant negative O-GlcNAcase in skeletal muscle, and showed that this led to muscle wasting. But we wanted to know is or does enhanced cardiomyocyte GlcNAcylation, is it sufficient to replicate a diabetic cardiomyocyte phenotype? So when you have a hammer, everything's a nail, and we got that TRE DNOGA mouse and crossed it with our alpha myosin TET-on model to make the cardiomyocyte specific system. So Brahma went in and tested it. It did induce, shown at the top is a Western blot with the endogenous OGA. You can see there is a feed forward loop of it. And then the dominant negative is an EGFP fusion for tracking purposes, partly to get at some of those localization studies. So it's a little bit larger, but there is indeed enhanced total cellular GlcNAcylation. And consistent with Brahma's initial hypothesis, GlcNAc, independent of everything else that goes on in these imperfect diabetes models or everything else that could be going on with enhanced glucose delivery like signaling and PPP, et cetera, it was still sufficient to suppress BDH and ketone body metabolism. So this led us to conclude that part of the disruption in total cellular metabolism is directly driven by GlcNAcylation, and that this could not only involve this lack of switch from glucose to fatty acids, but also a suppression of the ability of the heart to use ketone oxidation. And this was confirmed across, because none of these models are perfect, we just tried to look at as many as we could, 4 different mouse models and in the human diabetics. Yes.
Have a phenotype?
What's that?
Was there any phenotype?
That's the next question. Great lead in.
Sorry.
So just like our astute audience, my astute second postdoc was observing and say, well, okay, if this is sufficient to change metabolism, what else might it be doing? So what Jenna did was took these mice, turned them on again, but now instead of just turning it on for the 2-week time period, what goes on when this is sustained? So we picked 2 and 6 months, partly because we were doing echo serial cardiography, and that's when we were kind of seeing, well, is there a phenotype? And the answer is yes, there is. The mice develop hypertrophy, which we did not see in the GLUT4 mice. So even though enhancing glucose uptake is probably hitting a lot more pathways, either to the degree we're enhancing it or something else that's unique about modifying this dominant negative OGA or the removal of GlcNAc is affecting the hypertrophy pathways. And this is one caveat about this model. So since it's a dominant negative, we're affecting its removal, not enhancing its addition. Several other groups have overexpressed OGT, and that leads to a very severe phenotype. But OGT not only can do O-GlcNAc transferase, it also has a protease activity. So by doing the dominant negative OGA, we could look at enhancing GlcNAc without those secondary effects. But the phenotype is a hypertrophy. Sorry, this is showing the O-GlcNAcylation. This is the hypertrophy slide, sorry. So when we harvest those mice, particularly in the females a little bit more, there is total heart hypertrophy. And interestingly enough, especially in the females, there's a right ventricular hypertrophy that we still haven't figured out. I mean, this is being overexpressed in all the cardiomyocytes, but unlike most models of diabetic cardiomyopathy, that's primarily left ventricular hypertrophy, we see both left and right ventricular hypertrophy. But where things got really interesting for us, and part of the reason why we went all the way out 6 months, is we do not see systolic dysfunction. So doing echocardiography on these mice, we do not see a decrease in ejection fraction. We see a modest dilation in the males, very modest in the females, but we do see diastolic dysfunction. And when we first submitted the paper, we said, O-GlcNAcylation leads to HFpEF, and the reviewers quickly slammed that down and said no and rejected the paper. So it's now currently under revision at a different journal. But we do see this hypertrophy with preserved ejection fraction, with diastolic dysfunction, with fibrosis. So everything else that's hallmark of a diabetic cardiomyopathy. And this is consistent with the metabolic changes that we would predict, this decrease in oxalase.
Think about the cause of fibrosis in those mice.
Good question.
Sorry, Jinko, which one?
What is the cause of fibrosis?
Fibrosis.
So for those in the back room, what's leading to this fibrosis? That's hopefully going to be the next RL1 because this is a cardiomyocyte specific overexpression. So we think, and one of the things, again, a lot of this is done in collaboration with the Chatham Laboratory, is either because the glucose this enhancement of O-GlcNAcylation could be occurring at a lot of different sites. So O-GlcNAcylation in this context is the cellular signaling. If we're impacting UDP-GlcNAc pools, UDP-GlcNAc can be used for any glycosylation. So we may be affecting surface glycosylation that may be recruiting something. We may be affecting some type of secreted peptide. We do have some candidates that we're looking at. Flostilin, if any, it's a secreted peptide that is induced in these models that might be signaling to macrophages to bringing them in. The reviewers fortunately did not ask that question. So we're not getting that for this resubmission, but we are brainstorming some of that for the next grant. But if anybody wants to collaborate on that or has any ideas, I'd love to hear them. Yes.
It's kind of a general question, but probably it's a good time to ask it. So hypertrophy, the number of myocytes is not supposed to change. So what happens that every myocyte grows in volume, and how exactly it occurs? How the volume and shape changes?
So that was also something we anticipated the reviewers asking for. So we have all the WGA staining, but the main thing they asked for was O-GlcNAcase activity. So Jenna's just been working on that, but we have the stains, we just haven't quantified it. Because one of the things, and this isn't the best image, I'm not a histologist, despite being in a pathology department, is we do see infiltration of other cells. So we do have to rule out that the mass change is not just that. But from the early WGA quantification, the individual cardiomyocytes do seem to be growing. And it is consistent with some of the transcriptional hypertrophy markers that you would predict, like the MHC alpha beta switch. Some of those are consistent with the pathologic hypertrophy. So, but all good questions, but we haven't been able to get into the nitty gritty quantification of how much of the mass increase is cell infiltration versus classic cardiac hypertrophy versus just heart growth. All good questions. Thank you. So we continue to look of what other of the changes occur that are consistent with the diabetic cardiomyopathy. And we see a progressive decline in oxphos complex activity. And we see, unlike again in the MICLU4 mice, that there is some also other changes outside of the mitochondria as well, such as citrate synthase being decreased. So this is a different mechanism than simply driving glucose delivery. But I think this gives us more insight into not only the potential pathologic roles of O-GlcNAcylation, but by seeing what its regulating independent of the hyperglycemia and the less talked about hyperlipidemia that also occurs in all these diabetes models, what its physiologic roles might be, because when we disrupt them, what's going on. So what we've gone on to do with this, again, all the phenotyping and some of the protein work is then to do the transcriptomics. And we see a very clear transcriptional change driven by GlcNAc, which would make sense going back to this original concept of a lot of the O-GlcNAcylation targets are going to be transcriptional machinery. So the way we're hoping to conclude this paper, which again, hopefully will be resubmitted the end of this month, is that O-GlcNAcylation, when sustained independent of everything else that occurs in the diabetic milieu, is sufficient to lead to a cardiac hypertrophy, a cardiac diastolic dysfunction with fibrosis, and metabolic transcriptional remodeling and mitochondrial function consistent or reminiscent of a diabetic cardiomyopathy, further suggesting a key role for GlcNAc signaling as part of the metabolic changes that occur in response to diabetic changes. So any questions before I change gears the last time?
So in that last transcriptomics
Yes.
study, did you find any, I know you said a lot of them are like transcriptional regulators. Are there any that are maybe involved in both metabolic reprogramming and hypertrophy? Like several of them do.
Yeah, so, and I brought a lot of extra data and I'm definitely for sharing more than holding back. I know I already squeezed a lot in here, so I didn't squeeze everything. The answer is
Yeah, like convergent.
yes. And for this audience in particular, I did look up, and connexin 43 is one of the regulated genes. So I don't think it's only on the metabolic side. So connexin 43 is decreased statistically significantly as early as 2 weeks in both males and females, and goes a little bit more down at the 24-week. So it's not only metabolism, it's just metabolism's my hat. So I need to give somewhat of a coherent story if possible.
In the clinic, like in hypertrophic hearts, is there any evidence for this pathway being dysregulated?
Yes.
Sort of like the-
Yeah, so in heart failure, and this is where it gets a little bit complicated in my mind as far as this difference between a diabetic cardiomyopathy and a non-diabetic cardiomyopathy, because GlcNAc does seem to be up in both. And this is where some of the controversy in the GlcNAc field, in my mind, swirls, because people like to think as robots in binary, good or bad. And it's not. So there's a lot of early work, particularly out of Steve Jones' lab, out of John Chatham's lab, on enhanced O-GlcNAcylation actually being cardioprotective, especially in the context of ischemia or in stem cell regeneration and things like that. So it does get iffy, but in overt heart failure, independent of diabetes, you can still see an increase in O-GlcNAcylation, which this is where John and I differ a little bit. I came at this from more of the original thinking of this being a nutrient signaling pathway. But what John has shown is nutrient deprivation actually enhanced total cellular O-GlcNAcylation tenfold more than glucose surplus. So at the end of the day, I think it's a stress signal. So it gets a little bit more nuanced than just glucose driving. I saw 3 hands there.
You got it.
As you pointed out, O-GlcNAcylation is being controversial in terms of its cardioprotective or detrimental. And it's really amazing that only 2 enzymes regulate O-GlcNAcylation. When you think about phosphorylation, there's different kinases to specify the signaling, but in OGA and OGD, just the 2 players, but needs to have some specificity to determine, so okay, so the activation of O-GlcNAcylation, increased O-GlcNAcylation is cardioprotective or detrimental. So depending on what proteins are targeted by O-GlcNAcylation, so how specific is pattern?
So I read one more of a evolutionary epidemiology study of why that might be. And it's postulated that O-GlcNAcylation actually predates phosphorylation, but it was so critical that mutations in that pathway were all lethal. So whereas phosphorylation got more nuanced because mutations found a way and different kinases came, so that it is just this ultimate stress sensor. So where the specificity, if I could answer that, I might get a Nobel Prize, right? So one of the things that John and I are doing are with some of the subcellular distribution are to see why is it going to certain places in certain conditions and who's it interacting with. So there's early evidence that OGA and OGT actually interact with each other. So then they would be poised to add or remove at the same time, but who are their other players triggering which it is in which location? Because in some of our, some of the immunohistochemistry that Jen has done more recently that I'm not showing is under some of these conditions, we get puncta. So what are those sites of key regulation and what's special about them, or what's maybe the scaffold protein bringing them together and regulating it beyond whatever nutrient sensing there may or may not be. But
Yeah, I had some curiosity. Given you're saying transcriptional reprogramming, I'm wondering to what extent these kind of changes can be imprinted on.
that's part 4. It's the final part. So I'll get to that. And that's actually what I'm most interested in. So I saved the best for last.
Fair enough.
With your experience, do you think that this OGT and the OGA targets are different? So I think OGT has more targets than OGA. So does it like how they?
Yeah, I think a lot of the targets are similar because a lot of this isn't stable modifications. A lot of these do change somewhat readily, but you're right. I think by doing this dominant negative OGA, it's not like overexpressing an OGT. And I think that comparing to some of those models, so one of our collaborators, Rebecca Ritchie in Australia, has been doing some of that work comparing OGT overexpression versus OGA and to nuance some of that. The biggest problem is doing the proteomics. So we've gone through, I'm not a proteomics, I know there's a couple of proteomics people here, so maybe we can figure it out, but we've gone through 3 different collaborators trying to get to good site-specific proteomics, and it's just really difficult, yeah. But good question, I don't know that answer. Everything I've done so far, I mean, the best I've got into sites or to target specific even is that 2D page. So
The one question is that's a question which I normally get when I'm presenting with this data is that when can you say that like the acute with your experience, can you say that acute level of O-GlcNAcylation is something which is with cardioprotection, and when it is like excessively O-GlcNAcylation is something else?
yeah, so I think I've even used that language I think we used that in the review. We backed away a little bit from that because what's acute? I mean, acute in the phosphorylation world, seconds, right? We're talking 2 weeks. So, and that's why we backed away from acute versus chronic. And the fact of the matter is we don't see the dysfunction until further along. So yes, we're seeing the hypertrophy, but there is adaptive cardiac hypertrophy as well. So even at the 2-week time point, which I would say is already getting into the biologically chronic states, it could still be adaptive. And what I would be really curious about is then making these mice diabetic. Because if they have the disruption of the signaling with the excess nutrients, that's where you get to the disease states. So, okay. So for the final part, so there's really 3 arms in my lab. One's PDK, which I'm not going to talk about at all today. The other is GlcNAc, and the final one's epigenetics. And this gets to the imprinting. And one of the reasons why we're interested in epigenetics comes from early studies in type 1 diabetic patients, the EDIC trial, which was started as the DCCTT, or the Diabetes Control and Complications trial, which was done several years, I think almost 40 years ago now, where they took a bunch of type 1 diabetics, left them on the then conventional therapy, or put them on what they called intensive therapy to maintain their A1C levels at a lower level. It was stopped on ethical grounds because those on the conventional group had much worse outcomes. So then they switched all the patients onto the intensive therapy, which is now standard therapy, and one of the reasons why type 1 diabetics should all be on insulin pumps. But fortunate for us is they continue to follow out those patients, and even 20 and 30 years ago, there were worse outcomes in those patients that were initially on the conventional therapy, suggesting some type of memory that was produced from that glycemic control. Getting to the nuances between type 1 diabetes and autoimmune disease versus type 2 diabetes. Similar studies were done in type 2 diabetics. This is a Veterans Affairs diabetes trial where again, they did tight glycemic control or loose glycemic control. And although much more controversial, there did still appear to be some attenuation in long-term outcomes of the more tight glycemic control. So what is epigenetics? Epigenetics to me just ties in everything into transcriptional control, stress related, diet related, pollution related, not only what happened to you, what happened to your parents and grandparents related. When I use epigenetics, I don't use it as a geneticist of transgenerational. I use it as more of just another component of transcriptional control. The lab now has projects in all three of the main arms of epigenetics. We do a lot of collaborative work in the non-coding RNA. I'm going to present now our data on DNA methylation, which is one of the first identified epigenetic marks. And then our most recent finally funded R01 is going to be looking at histone acetylation in those PDKs. But one of the reasons why I particularly glommed on to epigenetics was because of those clinical trials, but also because all these changes in metabolism affect all the metabolic intermediates required to confer epigenetic changes, including allosteric regulation of the enzymes to moderate these. And I'm going to speed up just a little bit so we can get to the data, including O-GlcNAcylation. So O-GlcNAcylation can not only affect transcriptional control on transcription factors like SP1, like I already introduced, it can also interact as far as the specificity question goes with some of the enzymes that mediate these epigenetic changes, such as DNA demethylation via hydroxymethylation. And it's now been shown that a lot of the epigenetic machinery can interact with OGT and impact not only DNA methylation and demethylation, but also histone modifications. So is glucose and specifically GlcNAc sufficient to confer gene expression changes? So we collaborated with one group in Italy to first test, does just changing the amount of glucose in cell culture affect this one epigenetic change of DNA methylation? So this is in HEX cells where it was treated with no glucose, very low glucose or moderate glucose. And this is a sound just
Yeah.
I'll talk quickly since it's talk loudly. Okay, so what Mark Pepin, my first graduate student, did with this study was just did the bioinformatics of the methylation and showed that a lot of the changes that would occur, remodeling that would see in the endothelial cells, such as VEGF and NOS signaling, could be directly regulated by DNA methylation, by changes in DNA methylation. So going back to the GLUT4 overexpression model, what Cheyenne Bakshi, my middle graduate student, then wanted to do is build off of some of the work I had started when I first got to, I'll just continue, and say, okay, what type of imprinting may occur from this glucose? So what we've done is induce enhanced glucose delivery for 2 weeks, and then remove the doxycycline and let glucose delivery return to normal. So here's just the Western blot showing the enhanced transgene, and that goes away by 1 week and is gone by 2 weeks. Then we performed the transcriptomics and showed that indeed there was a large subset of genes that were induced by glucose delivery that were still induced a couple weeks later. And there were several genes that were suppressed that stayed suppressed. And several of these correlated to changes in DNA methylation. So this is a genome-wide, not a whole genome. We did not have the coverage we had hoped for when we first went into it, is why this is still unpublished. But from the data, we were able to glean, similar to the cell culture studies, in vivo enhancing glucose delivery could lead to some methylation enhancement, shown here in yellow, and some demethylation, and some of that demethylation stayed demethylated in the normal glucose delivery, and some of the methylation stayed methylated. But then this gets to the workshop. One of the things in the human study was that we think this is altering susceptibility to secondary stresses, that this is an imprinting that leads to a change in the ability to respond to something that would happen in a human later in life, such as hypertension. So for that, we use the transverse aortic constriction model, or TAC. And shown here is flow of blood up the right and left carotid arteries. And in a sham operated animals, I don't think you can see the scale here, but the velocity of the flow is about the same up either artery. But then what we go in and what I'll be doing this afternoon is putting a surgically a metal clip in between those two arteries to mimic immediate hypertension. Now, again, just like the diabetes models, this is not a hypertension model. This is only pressure overload on the heart. It doesn't have all the systemic effects. But what you can see now is the scale changes here, and up the right carotid, the velocities going from 60 to about 100, and up the left carotid from 60 down to 40. So the reason I use this model since my pre-doc days, but what we were using it for in this case is that epigenetic signal of that past glucose delivery alter susceptibility to the disease state? And the answer is yes. It's subtle in subsequent data, but the initial cohort showed that even when glucose delivery was returned to normal 2 weeks prior to the surgery, several weeks, a month later, there's enhanced mortality in those mice consistent with the prior delivery. Is this only glucose driven? Since we also have the O-GlcNAc model, we wanted to see if this could be specifically by O-GlcNAc as well. So Samuel Chain, my newest student in the laboratory, is now doing the same studies where, again, we now enhance O-GlcNAcylation for 2 weeks, remove it for 2 weeks, perform TAC, and show that even after 2 weeks, 2 months later, there's exacerbated hypertrophy. And we now have all the transcriptomics from these cohorts, and then Cheyenne and Samuel are doing the bioinformatics on that. And if you have me back, hopefully I hope in a year's time, the data will be done, but if you have me back sometime in the future, we should have hopefully some of the answers to that. So I know we're almost at the hour. So I want to thank the people. There's a lot of data I wasn't able to present, but people that contributed to the papers that I referred to in both the human studies and some of the mouse studies. All the McLoop-Force work was started in a postdoc with Dale Abel, who originally generated the model for its McTag to look at insulin signaling of GLUT4 vesicle trafficking. And then, as I mentioned, John Chatham has been my partner in crime for the O-GlcNAc work at UAB. Jen Brahma, my first postdoc, who's now moved on to an independent research scientist position in England, did all the ketone body work. Jenna is now a staff scientist in the laboratory and really does everything molecular biology. And Cheyenne does a lot of the bioinformatics, following up on Mark Pepin, who did all the human work I didn't talk about today. And then Samuel's the baby in the group, now picking up all the leftover projects. And with that, I'll take any more questions. Thank you.
So in your proteomics, did you find any DNA methyltransferases of the O-GlcNAc?
So I kind of glossed over that. So it was a 2D page of isolated mitochondria.
Oh,
so we weren't
okay.
expecting
So you
to
did,
see
it was isolated.
So it was a very specific question.
Mhm.
And we started there, A, because I only had enough funding to do a certain number of spots, but B, because we saw the defect in the isolated working hearts.
Okay.
So we figured since it was a defect in oxphos, we'd start with the mitochondria.
So now are you gonna do it?
Well, now I need to get a new grant for that.
Okay.
Yeah, so I'm hoping if this paper gets accepted, we're going to go in for the renewal to ask some of those questions.
I have a question about the ketone body utilization pathway and excessive synthesis pathway. So the random cycle is established and famous describe the crosstalk between glucose and pathways and but when it comes to excessive synthesis pathway and the ketone body utilization, what's the key to talk to pathways? So in your data, BDH1 was downregulated. So what's the signal from the excessive synthesis pathway to ketone body utilization of BDH1?
So, and that's part of what Brahma was trying to tease out. And I think there is some allosteric regulation. I think there is some Randall effect work, like metabolic signaling going on. the way we kind of concluded that paper, if I remember correctly, that we actually put in the paper versus what we discussed, was some of the more direct regulation that GlcNAc was working through some of these transcriptional pathways to decrease BDH expression so that there was some crosstalk there. I don't think that's the only case. Even going back to the GLU4 overexpression, I think part of the reason why the heart's decreasing this in excess nutrients is the heart is evolved to respond to these, but not in chronic states. So when you increase that signal and keep it up, it starts hitting everything. Because I showed you transcriptional regulation, post-translational regulation, and epigenetic regulation, all impacting oxphos. So I don't think it's any one, I think it's a multi-pronged death.
Another matter of curiosity, and I'm ignorant of this field, sorry. The problem with human populations for the last 100,000 years hasn't been excess food, it's been, not being able to find enough food. And so, presumably human populations have sort of cycled through times of famine, et cetera, et cetera. And I just want to kind of curious that, particularly the last day of the imprinting, that, potentially something could be passed down to the next generation that would affect their ability to undertake, in this case, cardiac metabolism. And I just wondered whether you'd think about this in a broader picture.
So there's a lot of great work not done by me in that area. There's several groups in Brazil that have looked at that. There's a lot of human population studies going back to some of the famine work, both, the Holland potato famines, a lot of the famines that occurred in China after the regime change that led to long-lasting effects through transgenerational epigenetics. And this gets to the multi-pronged death. So everything I'm looking at is more the Q changes. Some of these are passed on, and there's some really nice work showing giving a high-fat diet, pre-diabetic state to either the male or the female impregnated, and then switching to normal chow still leads to metabolic disturbances later. And then stress is a big thing. In human work, we're seeing great racial disparities that we've then connected to socioeconomic disparities. So the stress component, but all of that in human studies, it's too long to get to. So I know in the literature, there have been the largest mammal is some swine work regulating DNA methylation that's shown changes out 3 generations. In the C. elegans field, there's some microRNA work that has shown epigenetic memory out to 30 generations. So yeah, so then the question is, if it can be that long lasting, how do you reverse it?
Right. Yeah, and how does the energy flow over multiple generations relate to that? Provide advantage.
Especially when you're starting at one cell, because that's the thing that always perplexes me about these methylation changes, because everything I've shown you has been whole heart. We know that left ventricle and right ventricle vary. We know that cardiomyocyte versus macrophage vary. But if it's starting from one cell, how can it confer memory to the left ventricle or to the kidney or to the pancreas? And my hand waving response to that is the microRNA that are floating around with it that are signaling to some of the other epigenetic signaling, but I haven't figured out how to test that yet. Yes.
Wow, a lot of data.
Sorry. Let's go.
Put that over the time course of several weeks, right, in your mouse models. But I'm just wondering, almost all, a huge amount of the ex vivo heart work that we do with any sort of glycogen prep, we almost always feed the heart just glucose in those preps. How quickly do you think some of these changes might be happening, and how much of some of the canon of ex vivo heart literature might be impacted by these changes, these sudden and acute changes in energy substrate?
I agree entirely, particularly for muscle cells and particularly for cardiomyocytes, because essentially in a healthy heart, you can get whatever answer you want by which mix of substrates you're given. I think in the diseased heart, it's going to be more nuanced because I think there is this rigidity that I kind of alluded to in the introduction that occurs because of some of these modifications, but some of them would be occurring in the matter of minutes. for the post-translational modification. I think some of these transcriptional and definitely the epigenetic ones are going to take days, weeks, months, or even longer. And I think especially for some of the epigenetic ones, it's probably not the cardiomyocytes because they're not they're not dividing anymore. Now their gene expression's probably changing, but again, that would be longer term. But I know back from my thesis work on PGC, when we would overexpress it and study metabolism, we could basically get any change in glucose or fatty acid metabolism we wanted by what combination of glucose and fatty acid we gave the cells. So, and that's definitely true for ex vivo as well.
Questions?